So I would like to call this special seminar to order. Before I get started, I just want to introduce myself. My name is Martin Nyachoti. I'm a professor here in the Department of Animal Science, and I teach swine-related subjects, nutrition-related subjects, and do research in swine nutrition and management. I'm going to be the chair for this seminar today. So before we start, I would like to introduce or to let everyone know why we are here, know a little bit about this event, and then I will introduce our special guest speaker today, and then invite her to give the presentation or give a lecture. So this event is an annual event, lecture event, that recognizes and honors Dr. Tsang Kay Cheung, enduring support for the Department of Animal Science and Agricultural Research at the University of Manitoba. Dr. Cheung obtained his B.Sc, M.Sc, and Ph.D. in Animal Science from the University of Manitoba. His graduate training was in animal breeding and genetics under the supervision of the late Dr. Bob Parker. He was also awarded a Doctor of Laws from the University of Manitoba in 1991. Dr. Cheung returned to his family business in Hong Kong in 1975 and is currently the executive chairman, director of Herald Holdings Ltd. Dr. Cheung's many contributions include support to the TK Cheung Center for Animal Science Research, just out here, the National Center for Livestock and the Environment, the TK Cheung Reading Room, and the recent contributions to education and research in animal science. In recognition of his generosity and the commitment to education and research, the department established this annual seminar in his honor. I should also add here that the speakers that are invited for this event, they are all selected by the graduate students in the Department of Animal Science. And as faculty, we only do the invitation and the facilitation. So thank you very much to our graduate students for selecting the speakers and obviously for today's speaker. Thank you. So now let me introduce then the speaker there for today's lecture, Dr. Sara Place, who is an associate professor and expert in livestock systems sustainability with over a decade of excellence in academia, industry associations, and private industry. She joined Colorado State University's AgNext, a research collaborative focused on sustainable animal agriculture in August 2022. Sara's research focuses on enteric methane emission measurements and mitigation from cattle. Prior to joining Colorado State University AgNext, she was the Chief Sustainability Officer for Elango Animal Health, Senior Director for Sustainable Beef Production Research at the National Cattlemen Beef Association, and an assistant professor in sustainable beef cattle systems at Oklahoma State University. She received her Ph.D. in animal biology from the University of California, Davis, and a B.Sc in animal science from Cornell University. Sara is a native of upstate New York, where she grew up on a dairy farm. With that, I would like to invite Dr. Sara Place to take the podium and give us a lecture entitled Cattle's Role in Sustainable Food Systems. Please join with me in welcoming Dr. Place.
So to get started, just as a way of introduction, so I am currently at Colorado State University and a part of this research collaborative that we call AgNext. And so you can see our vision and mission that's on the slide here. This is a unique initiative by the university in terms of it's truly only focused on sustainable animal agriculture, and it does cut across multiple disciplines. So we have this mission to tackle this topic in a variety of ways. I'll share data later, specifically about the work that we do in enteric methane emissions measurement and mitigation, but we do work in multiple areas, and that is represented by this slide here of all the different faculty and professional staff that we have on our team. And really wanted to put them up here and recognize, especially with the data that I'll share later, these are the folks that are really key in terms of actually executing all that work and making sure that we are working together. But we have folks every aspect of sustainability, from economics to in terms of Dr. Nate DeLay and Dr. John Ritten, to more feedlot production and dairy production with Dr. Diego Manriquez and Pedro Carvalho. But then also we're focused on modeling. And so we're lucky to have Dr. Greg Thoma on our team as well in terms of life cycle assessment modeling. So trying to capture the whole range of sustainability and tackle it from many different angles. And of course, the people that actually make all the research work are our students, and want to give them a shout out here in terms of especially the data that I'll share later. They're the ones doing the hard work of actually collecting that information, and including, as we speak, today is actually a warmer day down in the States as it is up here, but the last week has been pretty chilly for them down there doing a lot of data collection. So really appreciate all their efforts always. So to get started, I chose this title of thinking about cattle's role in a sustainable food system. And one first thing to think about is, well, what is a food system, right? There's many different diagrams out there. I think this is a nice one, though very detailed and quite small. But the key thing is to look at those main components of what might make up a food system. Everything from really how is production taking place and the interaction of those biological systems with the environment, and thinking about the human environment and the economic systems, right, that also are a part of those sustainable food systems. Everything from the producers themselves to, of course, the entire value chain or the entire supply chain. And that's definitely important when it comes to sustainability in terms of commitments or expectations from more of the consumer side of the supply chain and how that can influence production practices all the way down to the farm or ranch level. And then, of course, we can think about that also being influenced by the demand of consumers themselves. But when we talk about sustainability, there's many players in that space, right? From non-governmental organizations to financial institutions. A lot of folks have put different pressures on the industry to do different things when it comes to sustainability, or have that expectation of reporting. And in the case of environmental sustainability, mitigation of negative environmental impacts. And so when we think about sustainability more broadly, we usually like to use this Venn diagram model of thinking about those three domains of sustainability. So from economic to social to environmental, and knowing and understanding some of these items that are listed here are not necessarily exhaustive by any means, right? There's many issues that can fall under each one of those domains. And really our challenges come in the places of those intersections, right? Those nexuses between the different domains. How do we manage trade-offs? How do we quantify those trade-offs? And hopefully in some cases turn them into more synergies in this space. In terms of an actual formal definition of sustainable food systems, that would be listed here in simplified form from the UNFAO, right? A system or a food system that provides food security and nutrition for all in such a way that we're also paying attention to economic, social, and environmental bases, right, to generate food security and nutrition for future generations, right? So that kind of historical context of meeting the needs of the present without sacrificing the ability of future generations to meet their own needs, right, from a UN sustainable development standpoint. What makes all of this challenging, and I'll go into the rest of the presentation with quite often a very narrow focus on just the facts and the data, but what makes sustainability always so challenging is there's a lot of value judgments that are infused in the whole topic, right? And often we can think about that as summarizing it and saying it's not just questions about what are we doing, but rather should questions. How should we be producing food, right? What should we be eating? How should animals be raised? And as soon as we use those should questions, right, we've departed the land of pure objectivity and we've infused a lot of value judgments. So there's never necessarily a pure answer in terms of what is right or what is wrong. Rather, we're always working on that continuum of better or worse. So how do cattle fit in this system? So we'll go through some of these main areas, definitely not exhaustive, and hopefully have a good discussion about other perspectives and other questions in this space. But to start, it's always important to think about that sustainable food system. The provisioning of food, the provisioning of nutrition is always a good place to start, right? And thinking about where cattle fit from that perspective. So if we look at it from a very macro level and just at the full global scale, how much nutrition, right, or from macronutrient standpoint, is actually coming from our bovine systems, right, for beef and for dairy. So what we're looking at here is information from the UNFAO stat of how much of our calories, right, our energy, our protein on a crude protein basis and fat supplies, just in aggregate, not accounting for losses and waste, is coming from beef and coming from milk. Milk is a little bit higher, but also there's no way to separate out in the FAO stat database that I can figure out our sheep and other small ruminants, right? So just keep that in mind. That includes all milk. But what's clear here, right, is when we think about animal agriculture, and this is universally true, our calorie value is relatively small in terms of the contribution, right? But where it matters more is getting into our protein, our fat, and then as I'll expand upon, really our micronutrients is where those animal source foods, those nutrient-dense foods really show up and matter, right? In aggregate, we're getting much more of our calories from other sources. The other key component of this is, while global averages are always useful and interesting to look at, the variation around the world is substantial, right? So what we're looking at here is just from that same database, the availability of bovine meat in kilograms per year. So availability, again, meaning just at a macro level, accounting for imports, exports, how much is just available in the country per person per year. And we see a tremendous amount of variation around the world. So using, again, a single number is not necessarily reflective of the lived experience of everyone in terms of how much is available and how much it would be contributing to their actual nutritional requirements. And then we can see the same for milk availability, again, with that caveat that this would be inclusive of all lactating ruminant animals, right? For the most part, ruminant animals contributing to this. And we can see that similar pattern, less of a high contribution in South America, right? But in the most part, the Americas and the global north tend to have, right, higher availability as with bovine meat. So back to this other more complicated view of things, and I think this is always the bigger picture when it comes to nutritional security, not just with cattle, but with all animal source foods. So this is a pretty information-dense slide, so I'll just kind of walk through it. At the top, this is a statistic that's always a little bit heartbreaking, right? That we have nearly 1 in 4 children in the world that are stunted, right? So not necessarily meeting their full potential, both from a physical growth standpoint or a cognitive development standpoint. That's not only caused by the lack of nutrient-dense foods such as animal source foods, but a key component of it is that contribution, right? And the other part of this slide is a nice report that came out from the GAIN Alliance, looking at this just macro-level relationship between the percentage of kilocalories in a nation's food supply that come from animal source foods. That's what ASF stands for here. It's always an unfortunate acronym, right? Because for a while there, ASF was definitely African swine fever, so this was not a good slide to show. And then the prevalence of inadequacy of availability for these different nutrients, okay? Each one of these dots represents a country average. So ultimately, the relationship there kind of highlights that inverse relationship of we can drive down inadequacy, tend to drive down inadequacy when there are more animal source foods available within a country, right? Again, there's multiple things at play here. We have those countries also tend to be a lot wealthier, right? So there's many things that are moving in the same direction. But this relationship is quite interesting of about 35% of energy from animal source foods tends to lead to a very nutritionally secure, right, population. So keep that threshold in mind, right? 35%. How many countries on average are actually achieving that? Actually not that many, right? When we look around the world at that macro level, right? So here we have our animal source foods as a percent of the kilocalorie supply in a country, and these different colors corresponding. Again, most of Africa is far below 20% of animal source foods as a percent of their food supply. We have exceptional countries, right? Places where there are a whole bunch of ruminants because of the grassland situation, or a lot of fish, right? In some places, like in Iceland. But even in the United States and in Canada, we're actually below that 35% threshold, which is interesting, right? Partially that's the fact that, especially in the United States, we're awash with calories, right? We have enough energy. We're set in that standpoint, right? And unfortunately, some of the foods that we eat are not that high quality, are plant-based, right? So think Twinkies are a plant-based food, right? So probably not a good example, but that's one of those things to keep in mind too as you look at this ratio. So that nutritional security is important, right? So livestock in general important, especially cattle, in terms of especially providing those nutrient-dense foods. Vitamin B12, zinc, iron, those things are incredibly important, and that's where animals show up as more important in our food supply. But we also had to think about some of these bigger picture issues in terms of resource competition when it comes to livestock and cattle more specifically. And this is one of those things that often folks will bring up as a critique of cattle production, right? We're competing with other potential ways to produce food, potentially more efficiently, right? Depending on how we define that. And that matters for both the feed that's used directly and for the land use, right? And so when we look at some of the data that is out there, this is from a paper that's a few years old now from UNFAO authors, looking at land use and what is suitable or what is not suitable and actually has livestock or animals on it, okay? So as we look at this graphic, as we look at this distribution of land, there's a few different categories here, right? One that's important for North America is this dark green grassland suitable for crops with animals, right? And so this is always an interesting component of when we think about land use competition is most of all the North American Great Plains is highlighted as suitable for crops. One of our challenges, of course, when we think about this bigger ecosystem perspective is in many cases from a conservation standpoint, we're trying to prevent further, right, conversion of native grasslands to cropland production. So just because something is suitable doesn't necessarily always mean we want to convert it, right, from other environmental sustainability standpoints. Though a lot of that conversion is happening because of economic and climate change factors, right? So as we look at this, the key takeaway, even though I don't have these stats here, is that a lot of the land, particularly for ruminants and cattle, that is used for production is not necessarily in competition with other potential food sources, or in the cases of this dark green, as we see in parts of the world, we would have that tension there of what is the best use from a long-term sustainability standpoint in terms of the erosion and other factors that may be at play. In terms of direct feed food competition, there's many analyses of that question now, I would say, but I think one of the original ones that I've ever found was back in 1980, this chapter in a book for alternative strategies in food animal production. And one is a good example of these debates kind of just recycle continuously, right? We've been having the same kind of debates about feed food competition and the efficiency and sustainability of livestock production. It just seems today we're adding the layer of climate change, whereas in the past that wasn't necessarily as important. But as we look at this chart, a key thing that is the takeaway here is that these two authors, Bywater and Baldwin, were looking at this issue and critiques that were coming out of the 1970s, especially as population was growing. There was a lot of concern about we're going to have too many people on planet Earth, and so we need to change our diets, right? We hear those kind of conversations today as well. And so a lot of statistics were coming out at the time about how much feed was being used by livestock and that it would be better put to use if we ate those plants directly. So this quote here is from this article of a fundamental issue in terms of efficiency is how it's actually expressed, right? The ultimate concern is total human food production. It only seems rational we should calculate animal efficiency in terms of inputs of human edible energy and protein and not total feed, right? So this is kind of that perennial challenge when we talk about feed conversion and sustainability is oftentimes folks are using just pounds or kilograms of dry matter, right, and conversion thereof to make that argument. So as we look at this chart, we can kind of see the differences there that this makes. So over on the left-hand side here is just kind of this macro total in, total out in terms of dry matter, and then also calories and protein not taking into account the actual human edible component versus the right-hand side where we take into account is this energy or is this protein source actually potentially human edible or not. So if we just zoom in and compare our cattle products or our milk here, right, we can look at the total inputs, the total returns, and this percent would say if we don't account for the human edible components, we're only returning 23% of the energy and roughly 29% of the protein in this situation. But if we take into account that much of what these cattle or other ruminant animals are consuming is not necessarily in direct competition with human food, then our returns go above 100% for energy and protein, right? And again, that's just accounting for things like forages, right, or byproducts that are not directly human edible that would be in those animals' diets and accounting for that ratio when making that adjustment to the conversion, right? So again, this just highlights that complexity, and we can expand upon that further if we use this example here. And this is a good example of also, when it comes to sustainability, those value judgments and the complexity of comparing, right? And ultimately, which metric you choose can tell a very different story, okay? So what we're looking at here is comparing beef from a grain-finished full life cycle perspective in the US to broiler chickens to pork. This is a UK and dairy, 2 UK examples from this Wilkinson paper. So the first column here is just simply our pounds or our kilograms of dry matter per kilogram of total live weight gain or for total solids in the terms of milk production. So as we first make this comparison, right, and again, that kind of historical perspective of just what goes in, what comes out, our cattle systems, right, at first glance appear to be a lot less efficient, right? And that would be something that we all know and understand, right? We're feeding those microbes first. We have higher maintenance energy requirements, et cetera. And so those ratios look higher for those animals as compared to our monogastrics. And again, keep in mind, this is a full life cycle perspective for beef, so we're taking into account the cow-calf and the stocker portion as well. If we adjust for what these animals are actually consuming and how much is human edible, we end up in this middle column, okay? So here the ratios across all 4 systems look relatively similar, which is always interesting. The actual amount of human edible inputs, mostly here in terms of corn and soy, right, that we're putting into these systems, looks quite similar, right, across all 4. And then we get to this idea of human edible returns, but then I've added something on top of this, right? And so this is looking at not just human edible returns in terms of, say, mass of protein going in and out, but now we're looking at the quality of the protein as well. So using this DIAS score in terms of the digestible, indispensable amino acid score of both the product, of the animal product, and the quality of the feed going into the animal itself. So in this case, we're flipping it on its head, and a higher number is better, right? So above 1 would mean we're returning more high-quality protein than we're using in these systems. And you'll note for both beef and for dairy in these 2 examples, right, they're well above 1. Whereas our monogastric systems are less than 1, which would make sense because we're using more high-quality feed inputs into those animals, right? So as we look at this, which animal is most efficient? It all depends on how you define things, right? And that's one of those challenges in this space is that you can kind of pick and choose statistics to meet whatever story you would like to tell, right? My bias of being more into ruminants, I would like that 3rd column, right, to be where I'd focus. But as we'll expand upon and as we talk about things like greenhouse gas emissions, there's also a different relationship there, right? Where the more human edible feed inputs we use, we tend to make fewer greenhouse gas emissions. But that's one of those central tensions and trade-offs in sustainability. So pulling some of this together, and one of those bigger picture concepts with regard to the feed-food competition is also just thinking about where it fits from more of a circular system, right? And this is quite a simplified diagram, but thinking about all those relationships between cattle production and between crop agriculture, right? Our human food, biofuels, fiber production systems, and all those interplays of how these things are connected, right? Ultimately, what we really care about is hopefully strengthening those connections where we can and linking particularly things like manure back up to our crop production where we can. There's some great efforts down in the United States with USDA looking at manure sheds concept, right? And thinking about how can we better make sure our nutrients are where they need to be and we're not concentrating them in certain regions, but that becomes difficult. But ultimately, we can kind of sum sustainability and environmental sustainability up as we're trying to strengthen these connections and then not have as much leakage from the system, which is inevitable, but ultimately that would be our pollution. So think things like enteric methane emissions, reactive nitrogen emissions, emissions to water, right? How can we tighten that circle up and not have as many leakages if we can, right? Is ultimately kind of our goal. And just to sum this up, if we think about the bigger picture of sustainability, there's a lot of non-food things that we're also providing, even though cattle are part of a food system, right? So yes, we're providing nutrition for people, but depending on where we are in the world, we're also providing potentially biofuels, we're providing building materials, we're providing clothing, right? Leather for steering wheels of cars, right? We're providing draft power potentially, pharmaceuticals, heart valves for people, right, from cattle, obviously wealth, other ecosystem services contributions, and definitely cultural value, right, which is sometimes underappreciated, but even if you think about cattle and all the symbols that we have, even in our modern lives that are related back to cattle, it's a lot, right, everything from Red Bull drinks to any other, idea that you may have that actually there's a connection to cattle, right, so in that bigger picture standpoint of our whole societal system, there's a lot of connections there. That all said, and what I spend a lot of my time on is like sustainability is this broad topic, and yet oftentimes it gets simplified down to climate change, right? And thinking about greenhouse gas emissions. And ultimately, a lot of this, at least in the last several years, has been coming from the fact that we have so many different areas where the cattle industry is feeling a little bit of pressure in terms of reporting on their emissions, mitigating their emissions, different commitments that are related to their greenhouse gas emissions. So some of that is coming from consumers, different consumer groups, NGOs. Some of it is regulatory, not likely our federal government in the United States anymore, but at least state governments, right? There's still regulatory pressure, right? Carbon markets from a more voluntary and from a business perspective. And then interestingly, banks and other investors has been a really big pressure point for a lot of our producers down in the United States. So expectations that they report on their so-called Scope 3 emissions that these banks do. So that means everything in their portfolio, they want to know where their emissions are at. And so that leads to a whole knock-on series of effects of investments and trying to figure out what are those emissions and how can we mitigate those emissions. So if we take, again, a very broad picture and we start at the global level, and we look at the latest UNFAO data, this would show us kind of where we're at in terms of cattle production for both beef and dairy around the world at about 7.4% of global greenhouse gas emissions, or at least related to this reference year that's 2015. So the Sankey diagram is really breaking those emissions out from starting, of course, all cattle, but then looking at where are those emissions coming from in terms of the cattle type. So interestingly, beef and dairy are roughly split evenly around the world in terms of where those emissions are coming from. Definitely depends on where you are, right? In the United States, we'd have much more coming from beef as compared to dairy, right? About a 2/3, 1/3 situation. And then where the production system type that we're looking at, so very few of those emissions are coming from what are classified as feedlot systems. More of it are those systems where we have a mixture of animal confinement and grazing or strictly grazing systems around the world. And then the meat and milk distribution, but the key thing here is looking at, well, where are these emissions really coming from? A huge chunk of it is enteric fermentation, right? So the natural production of methane from the animal's digestive processes is ultimately the single largest source of those emissions, and a big reason why cattle have a larger footprint than our other species globally is because of that enteric fermentation piece. Other of these categories are obviously more relevant to specific countries than others, right? If we think about land use change, that's what LUC stands for like pasture expansion, that's going to be highly variable depending on where we are in the world in terms of what that contribution is. So if we just zoom in on that methane piece, so just as there's different amounts of bovine meat and bovine milk available for people in each country, there's also a different amount of methane that's coming from cattle around the world, right? And if we think about beef cattle, our big behemoth here would be Brazil, right? In terms of most of the beef type cattle in the world are in Brazil, and so correspondingly, most of the methane is coming from that country, right? And a good example here is we'll kind of expand upon, right, the impact of productivity. I'm going to be mostly sharing here emissions in total, right? But this is where that emissions per unit of beef will come into play, right? In terms of the difference here, say, between the United States and Brazil, we actually have fewer cattle and make less methane than Brazil, and yet we actually make around 5 or 6 billion pounds more beef every single year than Brazil, right? So truly, the number of animals you have in a country for the total amount of production is one of the simplest ratios that actually matters when it comes to greenhouse gas emissions. And that's where all the things that we do for animal management, animal husbandry, animal genetics really play into environmental sustainability. So we see a similar, very disparate pattern around the world when we look at methane that comes from dairy-type cattle. And here, instead of being Brazil, our country that it shows up the hottest, if you will, in terms of the most methane emissions would be India, right? So unsurprisingly, a major dairy producer for our own domestic consumption, huge amount of the protein and nutrient requirements being met by dairy cattle in India, and so a tremendous amount of methane that's actually being produced there too. Also highlights some of the complexity there, right, in terms of some of those cattle are more, from a religious standpoint, viewed as sacred, right? And so there's going to be more of those animals around as compared to other countries, right? And so that drives those emissions as well, right? Those cultural, back to the cultural impact and significance of cattle. So if I take a narrow U.S. lens here, I'll drill into what are the life cycle assessment emissions. So this is more of that perspective of going into all the feed inputs, everything going into actually producing, right, those animals to the farm gate, right? What are the total emissions there? So here we're starting off, we're looking at beef cattle, right? Beef cattle production in the U.S. So our y-axis is the percent of the carbon footprint in the U.S., and this was work that really represents an average of 5 years in terms of the data that was collected as a part of this project. The big chunk of emissions is enteric methane, right? So 56% of the total beef carbon footprint in the US is coming from enteric methane, followed by nitrous oxide emissions that are occurring on land. A lot of that is manure inputs, urine inputs deposited on that land in terms of pasture, but also crop production, right, and synthetic nitrogen fertilizer inputs. Upstream is just referring to any of those inputs, say purchased fertilizer, things like that, that would have carbon dioxide emissions equivalents associated with it. But really the big chunk here is enteric methane. As a percent of U.S. emissions, about 3.7% of U.S. greenhouse gas emissions, okay? Where do those different emissions come from in terms of the cow-calf, stocker, backgrounding, and finishing phase? For this crowd, probably not surprising, most of those emissions are actually occurring on ranches in the U.S., right, on the grazing portion of cattle production. Whether we're talking about methane emissions, nitrous oxide, or anthropogenic CO2 in terms of, again, everything from driving ranch vehicles to energy use, right? Most of that is coming from our actual grazing portion of the industry. Upstream sources looks a little bit different because a lot of this is related to purchased inputs, including purchased feed. So unsurprisingly, the finishing portion, feedlots that are purchasing a lot of those inputs, shows up as a bigger percent for them, right? But overall, roughly it's an 80-20 split of 20% of those emissions occurring on feedlots, even though those feedlots are often accounting for 50% of the carcass weight gain, right, of the animals themselves. So that ratio there is definitely important. And just highlighting at least the demographics in the US, right? If we think about why those emissions are occurring from the cow-calf and stocker phases, right? Well, one is just that we have more animals in that phase at any one time in the United States, just as we would here in Canada, right? And the majority of what these animals are consuming is forages, right? And we know eating higher forage diets means more methane per unit of dry matter, and so we have more methane from a standpoint of the diets that those animals are consuming, and just more of those animals, leading to more of that methane coming from that part of the cycle. I've just highlighted up here, right, one of those central trade-offs and tensions as we have all this interest in trying to reduce methane emissions. Yes, we have more methane per animal in those phases, but a lot more logistical challenges, economic challenges, in how you're actually going to feasibly mitigate methane emissions in those systems, right? Really hard to feed a feed additive in many of our grazing systems in the United States, where sometimes ranchers are not even seeing those animals for months, right? Versus in the feedlot, where we have a lot more dietary control, many more options, right, to potentially deliver solutions, especially in rumen solutions, but less methane overall that's actually coming from that sector. So if we zoom in on dairy, so same situation. This is U.S. dairy from a life cycle perspective, all the inputs to the farm gate, so not including processing and all the way to the consumer. Again, enteric methane shows up at the largest proportion of our carbon footprint, but at a lower percent, about 43%. Here, the terminology changes a little bit. Resource production is similar to those upstream sources in the prior LCA. Feed inputs, purchase inputs, fertilizer inputs, things that are purchased would be the resource production here. And then manure methane on average in the US, and that's a key caveat, on average is about 20% of the carbon footprint of dairy. So this work from Al Rotz would say that about 1.5% of US greenhouse gas emissions come from dairy cattle production. But in terms of a total amount, right, about 100 teragrams or 100 million metric tons obviously exceeds some national inventories of greenhouse gases, right? So that's always important to keep in mind the percent versus the total emissions difference there. But this manure methane piece is really key. So as I kind of alluded to, this is a US average. This is quite variable. On some individual dairies, manure methane in the United States represents a bigger chunk of their carbon footprint than enteric methane emissions, okay? And ultimately, a lot of that is coming from how the manure is managed and the climate that that dairy farm is in. So this is information from another life cycle assessment from Dr. Thoma's work back in 2013. This graphic here, what we're looking at is greenhouse gas emissions expressed as kilograms of CO2 equivalents per kilogram of fat and protein corrected milk. And these different colors correspond to these 4 different buckets of emissions. So on-farm fuels down at the bottom in black, enteric methane in blue, in this kind of teal or tan color is the ration production or feed production, and then purple here is this highly variable part of manure management. So ultimately, I just want to highlight here, if you look at each one of these slices on this graphic, these are just simulated operations that were done in this life cycle assessment. So we have simulated operations with quite low carbon footprints, and a big driver there. Some of these other emissions change, but a big driver in the variation is this manure management piece. So ultimately, farms that are located in warmer climates, so say California or Arizona in the United States, and that have anaerobic lagoons to manage their manure, tend to produce much more methane than operations where their manure management is taking place in a colder environment. So that's always helpful in terms of knocking down microbial activity in the methanogenesis, and places where that manure gets exposed to air much more often. So as I've kind of called out, if you're still a smaller dairy that's actually doing daily spreading of manure, or you're grazing, that manure is exposed to the air much more and has a lot less opportunity to produce methane gas, and that often leads to a lower total chunk of emissions coming from that segment of production. So I'll transition now, kind of in the final chunk here, to a little bit of the work that we do at AgNext. Specifically, as I mentioned, right, we have folks doing work from animal health to modeling, et cetera. I'll just focus in on a little bit of the work that we've been doing on enteric methane emissions. So to summarize at a very high level how we try to think about the work that we're doing in enteric methane, the first would be thinking about trying to better baseline and create better prediction models in this space, and especially for cattle systems that haven't had a whole lot of measurement in the past, our finishing systems, and then also grazing systems as we continue to accumulate data in that space. We care what goes in the animal, whether it's different dietary or additive interventions, and then what we're really interested in as well is the genetics of the animal themselves. So we're just in the midst of trying to collect a lot of phenotypes of animals and thinking about potential future work in rumen microbiome manipulation, thinking about different more targeted additives in addition to the ones that are currently just getting out on the market. And I've highlighted here, we often have situations where animals are genetically similar. We're feeding them the exact same diet within a pen, and we can see roughly 30% differences from animal to animal. Which obviously makes it fun to try to find dietary differences, right? But then can be very interesting of saying why. Why are these animals so different? What is happening with the rumen microbiome that is making them so different that we see that range of methane emissions? So most of this work takes place at what we call our climate smart research pens in north of Fort Collins. So this is just kind of an overview shot of that facility. And it was a unique time where we actually had Brahman cattle and Angus cattle and some Herefords in there, so we had kind of the rainbow of animals in the facility, right? So the way these pens are set up, we can house up to 300 animals at once. And at the front of the pen, we have 5 smart feed units and 1 green feed in each one of these pens. So we can get individual feed intake and individual methane, CO2, hydrogen and oxygen fluxes on these animals. And then we have a facility where we can get individual body weights. So that allows us to get a lot of high resolution data on individual animals and again, look at some of those different research questions. So many of you may be familiar with the green feed system. This is a commercially available way to measure methane, right? In essence, it is a feeder where the animal can come up and visit the feeder voluntarily. This is always, when I'm showing this to non-ag audiences, have to put the disclaimer out there, their head's not stuck in here always, right? They come up, they voluntarily visit, they get some bait feed. We've typically used alfalfa pellets, which has worked in cross diets to actually attract the animals. And so while they're there consuming that bait feed, their emissions, that anything they respire, eructate, will be collected, right? In that system, and the gas analyzers are below the feed tray here, right? So in essence, it works with the principle of we measure the concentration in the background air, we measure what comes out of the animal, and we precisely measure the flow rate of the air that's collected in terms of the mass flow rate, and then we can get an emission rate from these systems. Basically, what we're getting is a spot sample each time the animal comes, right? So as I'll expand upon a bit later. In terms of what the raw data looks like out of these systems, I always think it's interesting in terms of looking at the pattern of eructation that you can observe with these systems. So this is, again, just the raw data from the interface of one of these machines from earlier this month of some bulls that we have in this current facility. And so what we're looking at here is just the raw concentration of methane on the y-axis. And you can see the animal enters the feeder, right, to get their snack, and then we see this pattern of eructation events that can be captured by the system, right? So it is almost like clockwork of roughly once a minute. That animal has that eructation event, and that gas is pushed out of mostly their nostrils and their mouth, right, and then it gets captured in the system. What's always interesting as well is even though we have these eructation events, the emissions, right, are not going down to background levels, right? So basically the animals are respiring methane all the while as well, right? And so that the concept there, again, we take that total area under the curve, and knowing the mass flow rate, we can get an emission rate, a spot sample emission rate every single time the animal visits the feeder. So one of our first questions was, despite these things, these green feed units having been around for 10 plus years, there's a lot of questions of still what is the appropriate number of spot samples for the animal, right? So not the most exciting research, but definitely important if we're going to do things like genetic evaluation. We need to know if we're actually collecting a good phenotype or not. What is a good phenotype when it comes to methane emissions? So this experiment here, we looked at collecting data from a few different experiments. In total, the animals that were eligible for this analysis were 213 crossbred animals fed a more high forage diet, 169 Angus steers, and then 64 Angus steers and 2 finishing diets, these last 2. And ultimately what, again, our great folks did, what Juan mostly did in terms of this analysis was looked at the gas flux for animals from the first 100 visits with either 2 or 3 minute duration length. So that's always another question of how long does the animal actually need to be in there? Does it matter, right, in terms of the final data? So we looked at those animals with 100 visits and then basically looked at how well looking at 5 visit interval chunks correlated with a full 100 visits, meaning visits 1 through 5, visits 1 through 10, visits 1 through 15, hopefully that makes sense, so on and so forth, forwards and backwards to see what was the actual relationship there between the overall mean and those subsample means, okay? So the kind of takeaway of that paper is kind of summarized in these 2 charts. So what we're looking at here is the visits that are 2 minutes or greater in length, or 3 minutes or greater in length. And here, just for simplicity's sake, just looking at the forward ranges, so again, thinking that mean for 1 through 5, all the way to 100, right, in both situations. The lines here are corresponding to the dashed red line and the dashed blue line are the Pearson and Spearman rank correlation coefficients. And then the dashed green line here is the residual variance. So unsurprisingly, as we add, right, these visits up and we get closer to the total 100, our residual variance declines, the repeatability increased, and our correlation gets higher, right? If we use this 95.95 threshold for both our Pearson correlation coefficients and our Spearman rank, we end up somewhere around 55 spot samples to collect a representative mean, right, or 60 if we used our longer period, right? So we feel good, and we try to use this as our threshold with experiments going forward of trying to accumulate at least 55 visits from individual animals to get a good representative sample, right? And this would be in line with what others have recommended, but kind of a highlight of we probably as a community of folks that use these devices need to come together and say, you know, what is kind of an overall recommendation, right? So just a summary here of some other folks that have tried to do similar type of analyses using different approaches, right? We have ranges everywhere from about 30, right, to the highest of, we kind of repeated this analysis for a grazing data set up to 70 visits, right? So ultimately, a lot of this is just driven by the type of animal, the growth stage of the animal. Unsurprisingly, methane is related to feed intake. So if an animal's feed intake is rapidly increasing over the experimental period, then you know this can change, right, the answer that you get to this question. But ultimately, this just highlights we often are not reporting this type of data in these type of papers, and we probably should be, right, in terms of comparisons to others, in terms of how many visits does each animal have from these green feed machines. So we also have had other questions and digging into this deeper, and this is just kind of the initial analysis of if we're going to talk about this potential for genetics or even just classifying animals, are animals that are low, so-called low emitters, right? First of all, how do we define that? Low emitters on high forage diets, do they remain low emitters if we feed them on a high energy finishing ration, right? So this is just one subset of data that we tried to start answering that question, where we looked at animals where we measured them twice in their life, where they were eating a higher forage backgrounding diet. These animals were a part of a different study where we either implanted or did not implant them in the finishing phase. So that's part of what's in this analysis. And then we classified these animals as low, medium, high emitters in a few different ways. So fairly straightforwardly, just purely from methane production, methane yield, methane intensity, And then residual methane emissions, which we'll kind of continue that analysis on now, of different ways to express residual methane, just as you would look at, say, residual feed intake. So in this particular analysis, we're looking at the midpoint body weight of the animals in terms of metabolic midweight and the average daily gain of the animals. The dry matter intake of the animals. And this is going into our prediction, and then the residual from that prediction is what residual methane is here. So we used our 55 visit threshold in 2 minutes or greater. And in terms of the answer, do these animals re-rank themselves? The answer is yes, they do, right? So that's kind of the summary of, unfortunately, they do, right? It would be a lot easier if those animals that were low emitters on high forage diets were just consistently low emitters on high grain diets. But what we're looking at here in terms of this graphic is the proportion of steers that maintained in the same category, so in that high, medium, low, for the four different ways that we classified emissions. So methane production, methane yields in terms of methane per unit of dry matter intake, methane per unit of average daily gain in methane I, and then the residual methane emissions. Okay, so between 35 and 83% of the animals re-ranked in each one of those categories. Okay. We also just looked at simply the relationship between methane emissions of those two different phases, right? So here's just our regression analysis of methane during the backgrounding phase versus in the finishing phase. We also looked at the Spearman rank correlation coefficients, right? Depending on how you categorize those, some would say those are kind of moderately correlated, right? But ultimately, that's a significant portion of the animals that are not maintaining the same rank, right? So for our conclusion, ultimately, we assume we have to actually collect those methane phenotypes within the production system that we want to influence. And ultimately, if more methane is coming from higher forage diets, we maybe care a lot about that and make sure that if we put any selection pressure on emissions at that phase, that we're not negatively impacting all the other economically relevant production factors in the whole system. And so we've done this type of work, which we're lucky to do, of measuring animals in confinement on high forage diets, grazing, and then looking at their emissions in feedlot systems. So we're lucky to have that type of data. And the key thing here is just coming back to the earlier part of the conversation, right? This is that key trade-off when it comes to enteric methane emissions and efficiency, right, depending on how we define it. Our high grain diets, we've seen this repeatedly, of when we take an animal and we put them in the feedlot, we lower their emissions by 30 or 40%, but we're now using a lot more human edible resources, right? So what is the right answer there in terms of that balance of greenhouse gas emissions versus feed food competition? And finally, we know things like production technologies can really make a difference, right? So just kind of a summary of the study where all we did was either implant animals or not at the end of the finishing period. And these were animals that had not experienced a growth implant in their prior production, right? And we can see the differences here in terms of average daily gain, dry matter intake, improving feed efficiency, improving total hot carcass weight produced. Given the market conditions at this time, pretty dramatic improvement in total revenue per head, definitely the implant paying for itself. And importantly here, a reduction in methane emissions per unit of gain within that finishing period when we actually implanted the animals, right? So we improved our revenue by 7% and reduced our methane intensity by 22% by just simply giving an implant, even though the animals' intake slightly increased, their methane slightly increased, right? Our gains increased more to drive down those emissions per unit of gain. Right? However, that's one of those things that's another trade-off, and back to the bigger picture when it comes to sustainability. Some of you all may have been following some of this that's been taking place, right? So of course, 3-nitroxypropanol, or Bovaer, is a feed additive that's now approved here in Canada, approved in the US for lactating dairy cows to reduce methane emissions, marketed by or produced by DSM and marketed by Elanco Animal Health in North America. And what's fascinating is what's happened in the last few months is the general public has become aware of Bovaer, and some of the reaction has not been very positive, right? There's been significant backlash, especially in the UK, in terms of kind of having, I would say, a bit of like almost an ick factor, or what are people feeding our cows, right? What is this chemical that's going into their feed? Why are we doing this to animals and changing their guts, right? And so if you haven't seen any of this and you simply search on any social media platform, Bovaer, right, you will see a lot of the commentary and backlash. But I think the key thing here is just that tension back to sustainability. We can come up with excellent technical solutions to address these very specific questions. But if there's not social acceptance of these technical solutions, then we're not really changing anything in the real world, right? I don't have the answer of how do we better address that, other than we should probably be doing a lot more of that up front before we get to, say in this scenario, a very good product that's at the end of 10 plus years of research that now faces a bit of an uncertain future in terms of its actual use on commercial operations, right? So that's a challenging situation, but one of the realities when it comes to sustainability is we have to circle back to that social piece. And then just to sum up, if we think about cattle and sustainability, this is a quote from Robert Hungate, but I think basically can be summed up if we think about the amazing thing that is cattle. We could probably pitch this to some of the Silicon Valley people, right, in a little bit different way of saying, hey, we have this amazing technology that can make plant-based meat. It self-replicates. It uses human inedible inputs. It's mobile and it doesn't burn fossil fuels, right? It fertilizes the soil and it produces this really high quality protein and other nutrient-dense foods for people, right, which is cattle. And so there are those trade-offs of the fermentation process of producing methane, but ultimately ruminants are quite amazing in terms of what they're able to do, and cattle are quite amazing, right? And so that bigger picture of how they fit in this sustainable food system is incredibly important. So with that, thank you very much for your attention. Happy to take any questions, and I really appreciate, again, the invitation to speak today. Thank you.
Thank you very much, Sarah. That was very, very interesting. So we do have some audience online, and we do have time right now for some questions. So for those of you who are online, Crystal, our communication specialist, is keeping track. So please send in your questions, and Crystal will ask those questions on your behalf. And for those of you who are here, of course, then you can put up your hand, and then I will give you a chance.
Can I just suggest that the people in the room come to the mic, just for the benefit of the people that are watching online? Otherwise, they probably won't hear you. So, and as soon as you're ready, I have a question.
Go ahead.
Maybe we'll start with this one, and then we'll get people in the room to come up. Great presentation. This is from Sage Hill Stables. For the grazing CH4 research, what type of supplements are they using in the green units so that it doesn't impact their diet drastically from what a grazing animal would be typically eating?
Yes, that's a great question. So what we are typically using for that bait feed is alfalfa pellets. So in terms of disrupting their normal grazing behavior, you do, right? And that's always the question of how can you measure methane emissions without disrupting their behavior? So some of that we are super interested in doing some more in-depth studies to say how much are we changing basically grazing distribution, right? Potentially total nitrogen excretion, right, from the animal by feeding them an alfalfa pellet. So we've actually run into that with some of the research collaborators that we have of they don't want to collaborate with us because we're going to disrupt their ecosystems work that they're looking at. So if we could figure out a way to measure emissions without having to ever affect the animal's behavior or give them an input that is new and different compared to what they would be exposed to, that would be excellent. But at least thus far, that's probably one of our best ways to measure methane emissions on pastures using these green feed units. Though I will say our rates of acclimation go down to about 7 out of 10 in terms of the animals that will actually visit the units when we're grazing versus 9 out of 10 or 9.5 out of 10 when we're in confinement. They have a lot more things to do with their day when they're grazing, and so they don't like to visit the machines as much. Yeah.
Okay, any question from the audience here? Okay, Dr. M.Sc.
Thanks very much, Sarah. That was really an outstanding presentation. I just wanted to, if you could comment a little bit about Bovaer. And so Bovaer offers some unique challenges because unlike many of our other mitigation strategies, it is neutral in terms of production efficiency, right? So from your experiences in the U.S., are you seeing Bovaer adopted in the feedlot scenario where you can actually formulate the diet with Bovaer in it to a significant extent? And do you think that would change substantially if we started seeing companies invest in offering some incentives for feedlots to be able to use Bovaer more extensively?
Yeah, great question. So in the U.S., it's fairly limited use thus far, and it's only approved by our U.S. FDA for lactating dairy cows, so not yet in feedlots. But yeah, the lack of a productivity impact has been kind of significant in terms of thinking through, well, how will it actually get adopted? So a couple things there. One is we do have a couple simplistic calculators that we've made just as decision tools for producers to use any sort of generic feed additive and look at the basic economics to answer that question right off of like, does it make sense for you to use these feed additives? And those are called FACT tools, so we call them our feed additive calculator tools. And a key part of that calculator is, well, what is the carbon price you're going to receive? So to your question, the strategy thus far in the US has been trying to create more of a carbon inset market. So rather than an offset market where somebody is doing some sort of activity, reduces their emissions, it's sold to somebody outside of the supply chain, carbon inset market would be saying, say a Nestle or Danone, right? Somebody who is an actual dairy product company or a Starbucks, whoever it may be, pays an incentive for a producer to actually use the product. And those type of markets are going to have to develop for a product like Bovaer where there's not been a measurable, repeatable productivity impact, right? There's some inklings of it in some literature of saying maybe we increase total milk fat production or improve feed efficiency, but I think that that type of data isn't very strong enough to incentivize using it right now. So there has to be some sort of premium, whether it's a carbon price premium or a premium that's just paid per hundredweight of milk that's not even related to carbon, right? Something that it has to be there to actually incentivize its adoption. Yep.
Okay. Go ahead.
One more from online. This is from Jay Wetter. I need a quick refresher on where methane comes from. What percentage comes from burps versus farts? And I think the methane measuring tool you profiled would only measure mouth-released methane, correct? So how do you measure the back-end emissions?
Excellent question. Yes, always a good one. So we always say it's almost all the burps, right? So if we look at total methane production, it's at least a 90-10 split of most of it taking place in the rumen versus the large intestine. But even some of that methane that's produced in the large intestine, right, we assume it gets absorbed in the animal's bloodstream, and that's probably partially what's being respired, right, by that animal. So we feel confident we're capturing about 95% plus of the animal's emissions when we're just measuring the front end. If you want to do a full animal measurement, that's when you have to start putting animals into some sort of confined area, whether it's a whole animal chamber or systems such as at University of California, Davis, where it's basically a feedlot pen that's covered by a coverall building. You measure everything coming in, everything coming out, right? But without that, yeah, you're missing roughly 3 to 5% of the emissions that are coming out the back end of the animal.
Very good. Another question here, Dr. Chichek.
Thanks, Sarah, for the excellent presentation. So this is from an outsider's perspective, 80% of your emissions come from non-feedlots, right? Which is places we can't really have a lot of intervention. So what are the low-hanging fruits in that case? Mm-hmm. I understand 17% of your emissions are feedlots. Let's say NOP or something like that can be added with incentives and so forth. Is it genetics? What can be done on the big part of that curve?
Great question. Yeah. So it depends on where we're talking about, what country and those type of things. Ultimately, globally, I think still, and that would be borne out with other analyses, our biggest opportunity is just improving the efficiency of the overall system. So whether that's everything from the genetic merit of the animals, animal nutrition, animal health, thinking about reproductive rates is an incredibly important metric. Back to that kind of quick example of, say, Brazil versus the United States, right? Truly just the number of animals you have in a country versus the production is really, really important. So that's part of it, is improving those system level things at that phase. But we can try to get creative of, yes, genetics that's why we're so interested in it, because it's not a daily cost for a producer, but rather a selection decision you can make and create permanent cumulative potential change. So we view that as a very attractive potential option if we can find animals that check all those boxes, including lower methane emissions. And then there's probably ways to try to get creative with other solutions. So we're going to start a project in a couple months of looking at oxygenated water with nanobubbles and infused in water. Can we change the redox potential of the rumen in a significant way and potentially lower methane emissions? There's a lot of ifs in that project, but that's what we're calling our bubble project. So that might be something as well, right? If we can come up with in-water solutions, other things of the few places that you're going to interface with an animal, a vaccine, some of those type of things might be more bigger picture options in the grazing phase.
Okay, another question from graduate students in the audience. This is your guest. Yes, Janine, thank you.
Hi, thank you so much for coming. I really enjoyed your presentation. In your opinion, do you see quantifying carbon sequestration or biodiversity as a way to offset some of these for a grazing situation? Or kind of where do you see that going in the future?
Yeah, great question. So on the carbon sequestration piece, that's something that we don't do a lot of research on, though we have great colleagues at CSU that are very interested in that, and colleagues at USDA. And so there is potential on the carbon sequestration side to say, can we offset some of those emissions? So even all those life cycle assessments that I shared earlier, that's only the emission side of the equation. So there's no assumed carbon sequestration. However, it's also really hard to sometimes influence carbon sequestration given your environment and the production systems. Some of our collaborators with USDA that have a station located not far from Fort Collins on the eastern plains of Colorado have a long-term 70-year experiment going on of comparing grazing versus no grazing and soil samples going back that whole time, and find really no difference in the soil carbon for those two different treatments, right? That also means that grazing isn't negatively impacting soil carbon, which is good. But I think that's part of the challenge is in certain parts of North America, more of the eastern North America, where maybe we're converting cropland back to grazing, we probably have huge potential to store carbon in the soil. But in a lot of those places where we have more arid systems, not a lot of nitrogen inputs, not a lot of water inputs, really hard to store more carbon, right? So it all depends. To the other part of your question, though, yes, I think that other part of it is broadening the picture, right? It's very easy to get carbon tunnel vision when talking about a lot of this, right? And we spend a lot of time talking about methane. It's important, but it's just one slice of that bigger sustainability piece. And I think that's what's very important for cattle systems is making sure we look at the bigger picture, and particularly when it comes to grazing, right? Our bigger conservation and ecological goals we may have societally is really where cattle are important, right? In terms of maintaining grasslands and all the ecosystem functions that are coming from those grasslands, from wildlife habitat to water filtration, doesn't really get captured in a carbon footprint. Yep.
Okay, we do have time for about one or two questions. Krista, do you have another question? Is there another question from the people here? Dr. Newman?
Excellent question. Presentation, sorry. Yeah. Excellent question. So, yeah, I was thinking, from the ongoing research that you have over there, especially I was really interested in that, the re-ranking experiment. Yeah. So basically, have you ever thought about basically, because whenever we are talking about the genetic selections, the breedings, we are just looking at the animal itself. But there is good indication that we have heritability of the microbiome. So is there any chances you have looked at the offspring of those cattle, high producing, lowest producing? And I think there is a very good opportunity if we can show that the microbiome is also heritable, then especially for the cow-calf operations, because in the dairy we are basically separating them from each other, so that heritability is cut. But I mean, for cow-calf operations, that's a very excellent opportunity. Yeah, so I was just wondering what's your thought about this?
Yeah, so that's why we're very excited about this research, because it is kind of ongoing, and we're collaborating with our geneticist colleagues in the Department of Animal Sciences. Basically, the data we're collecting right now, we hope to aggregate 3 years together and then get kind of a heritability estimate of methane. At least according to last year's data to this point, it was a heritability about 0.22 for methane, right? So these are animals where we hope we can take a slice of the CSU Angus herd and then test this. Can we create a low methane emitting line, right, over the next few years and just see if there's an influence there, right? So, and that is a disclaimer, of course, those are all phenotypic relationships, not genetic correlations, right? I'm not smart enough to do that. I leave that to our geneticist colleagues. But yeah, that part is really exciting. And the microbiome piece, we're also doing work with some colleagues where what we have been doing mostly now is separating those animals by residual methane and then sampling our high and low. And we do see very distinct populations. So we're hoping to pull that thread a lot more as well and say, why do we have these high emitters, particularly in the feedlot, in the high green diets, right? It almost, what has happened, a repeated situation, is when we look at the relationship of feed intake and methane, it's quite strong on a high forage diet. And then as we get into the feedlot, that relationship kind of breaks down. And so that animal-to-animal variation just shows up more. And so it's like, what did we do to those animals that we have such a different microbiome? Is it genetics? Is it a treatment decision we made when they were a calf? We have no idea, right? There's so many questions there that are, I think, pretty exciting to figure out what is actually driving that. Yep.
Okay, we are almost out of time, but one final question then,
Okay,
Krista.
maybe this will be a quick one. Again, from Sage Hill Stables, just to follow up on the nitrogen excretion, there was a graph showing nitrous oxide as the second greenhouse gas of concern following methane. Does this consider the global warming potential of nitrous oxide, and is there much research looking at nitrous oxide from some of these management systems and diet changes?
Yeah, so to quickly answer that one, yes, that is, didn't explain that very well, but when we show carbon dioxide equivalents, we're accounting for the much higher warming potential of nitrous oxide compared to methane. And then, yes, that is one of those things we always want to pay attention to. If we're doing something to affect digestibility or any other microbial processes in the rumen, are we actually just excreting nitrogen more and causing a potential knock-on effect? So we're not measuring nitrous oxide right now. We hope to start via collaboration with some researchers that are basically, simple way to put it is shooting lasers on either side of our pen and looking at the emissions from the whole system. So we hope to start getting into that space of measuring nitrous oxide emissions as well.
Okay, very interesting seminar. And thank you very much for all the questions. We've run out of time. That's all the time we have.